Okay, so this next topic it's going to be pretty heavy. It's a lot of information. It unfortunately changes a lot of the past and how you've been thinking. Things that you didn't have to think about in the past, now it's like no longer just a gimme. The idea is that we're getting into the fact that when you read a problem in the past, you didn't really look at certain key details. Like you didn't look at the name of the person in the problem. You didn't look to see if it was a boy or a girl. They were equal because every assumption up until this moment was that the gene that we were discussing was classified as an autosomal trait. And that means it is a gene that is found on an autosome, which is a non-sex determining chromosome. So therefore, the autosome in a male and female are equivalent. So for every autosomal gene, a male in their atypical somatic cell has 2 copies of that trait, as a female has the same exact thing. So there was no difference between this is the universal symbol for male, universal symbol for female. So there were no differences between that. So anything that we have discussed when there was a genotype of a male that, typical Mendelian, we always had 2 letters represented either of them. It didn't matter that it was represented by a female or a male. So that, unfortunately, is now something that you start to look at a little with a different lens because you're going to start to learn about sex-linked. So a sex-linked trait is the concept of this lesson. And that means that it's on a sex chromosome or a sex-determining chromosome. And so we have 2 different types of sex chromosomes. We have X and Ys. And females are represented by 2 X's, and a typical male is represented by an X and a Y. Now, when we say sex-linked, that could be any trait that is linked to either of these chromosomes. But if we're really concerned about tracing it through a family and determining the probability of it being passed down, we don't really focus on a Y chromosome or a Y-linked trait because it's pretty much guaranteed that if a male has an affected gene on a Y chromosome, it is going to be passed down to every male offspring. That is what made them the male offspring, is because their father passed down the Y chromosome to them. So there's not really a chance. The only other thing would be that there'd be a spontaneous mutation, which we're not assuming that's occurring in this scenario. So the only ones that we really look at are the genes that are linked to the X chromosomes. So when we say sex-linked, the word linked means attached. So it's attached to a sex chromosome. In our cases, the most common example is going to be an X-linked trait. So there are Y-linked ones, but we will not be doing Punnett squares for that. So how does that differentiate? If this is a female and this is your standard typical male, then you recognize that there is only 1 X chromosome that a male carries in every cell. So for a trait that is linked to an X chromosome, they can only have 1 allele, given the fact that they only have 1 X chromosome. For a female, she will have 2 X's, and each of them will carry its own specific variation of that trait. So therefore, she will have 2 alleles. So nothing really changes with her. But the way we notate it, just like on all previous examples, we have to notate it so that people understand what we are discussing. So your common examples, we actually have 3 human sex-linked traits that you should probably recognize. The big 3, as I call them, are colorblindness, hemophilia, and muscular dystrophy. Probably in most common order, going from colorblindness is a very common example. So all of these are caused by the recessive trait. So if someone that is colorblind would have some variation of recessiveness. Hemophilia, which is a bleeder's disease where they can't clot their blood upon some type of injury. And then muscular dystrophy is the breakdown of muscular tissue. Again, the idea is that it's caused by a recessive. So when you're doing a key, you kind of can do a generic. Big C would be I like to, again, use the word typical. I don't like using the word normal. And then a lowercase c would represent colorblindness. And you could do that for all of these using their respective letters. If you want to do H's or M's, it's up to you. But technically, when we build the genotype, what is important is the idea that now you must understand who has the affectedness. Is the person a male or female? Because the first thing you do whenever you see something of the big three, or maybe they come up with a random one that is also sex-linked, but for an intro bio, they're probably going to pick one of these three, unless they leave the human examples, and I'll show you some non-human examples. But what you first do is you set up the basic sex chromosomes, so that's the female, and then here is the male with your little cross symbol in between. So what you would then do is you analyze the problem. I'm going to start very, very simple and say that this is a scenario where what you have is a female who is a carrier. And they often use the word carrier in sex-linked problems, saying that they do not suffer from the condition, but they have the capability of passing it down. So there are capital and lowercase letters used in here, so you have to be well aware of which ones are the dominant and recessive. So it kind of has a blend of some codominantness, but you don't interpret it as such. So this is a carrier female, and if we then say that, you know, to kind of get some variety here, she is mating with a male who is colorblind. Notice the male has only one X chromosome, so the allele always is linked in a superscript fashion to the X chromosome. There is never going to be anything attached to the Y, because the colorblindness, hemophilia, or muscular dystrophy are not found on the Y chromosome. It's a completely different chromosome with its own set of genes. So we're not going to put any letters up there. So here, you now have the basic idea of how to notate a sex-linked problem. So you would do this on a simple monohybrid cross. I'm going to separate the letters out. Kind of switch between mine. If I decide the female, I'll separate her gametes along the top of the Punnett square. Then I build my Punnett square. And then I'll put the male over here. Now I can fill in which letters I separated. Nothing on the Y. So then you come down here, and I have my X big C, X little c, X little c, X little c, big C, and the Y has nothing, X little c, and the Y. So that's your Punnett square. Now what is going to be different is potentially how you write you have never done this when we write out a ratio. The genotypic ratio, you notice that there are, everyone has a unique genotype, unfortunately. So this would be a 1-to-1 to 1-to-1 ratio. I don't think people would struggle with that. But the phenotypic ratio is where things change, because there's different ways to kind of approach it. I think teachers all have their own little unique style. But what you need to represent is you're not just looking for the phenotype of the colorblind or typical, but you're also looking to see if they are male or female. So what you have here, this square represents one, and sometimes they'll say, okay, she is a carrier, but that's not a phenotypic expression. We wouldn't know that she's a carrier. We would just represent her as typical. She can see color, and you can kind of put female. So it has to kind of include the sex, and that's what's different about this phenotypic ratio than you've ever done before. The next square I would have, this is a colorblind. If you want to abbreviate, I'm perfectly fine with that. That would be one colorblind female. Here you have one typical male, and then you have one colorblind male. So what you're noticing is there is a separation between males and females. Another way to do this, if you want, this could get, when you get to dihybrids that have a lot more potential outcomes, maybe you separate it out and you say, all right, females, and you kind of put a one typical, this would be the same exact Punnett square, to one colorblind. And then you have to do a males doing the same thing. One typical, one colorblind. So it's just another option. This method seems to work when you have a lot more combinations. So it's kind of how to approach this. There are two different ways. I'll accept either of them. So what does this do? If you're unaware, a lot of the question they might ask you is, well, it is commonly understood that females suffer less less sex-linked traits than males do. And it comes down to this concept that they're asking you to really explain. With a female, there are two options to inherit a dominant allele, which could then mask the presence of the recessive. But a male, since he only inherits one allele, whatever allele is attached to his only X chromosome is going to phenotypically be expressed. So it kind of comes down to an odds thing, and hopefully that makes sense, where you're doubling the chance here, where you kind of can increase and get a dominant allele. So if you think about these, you have to start thinking about who do they get their X's and Y's from. So that's a big understanding. So if I do that on paper, if I have a male that is colorblind, who did he inherit this X from? So this is where understanding, all right, he has a mom and a dad. The dad passed down his Y chromosome. So you start looking at inheritance patterns and say, all right, well, mom is responsible for this allele. So kind of any boy that you know is colorblind doesn't get passed down from, because it's a sex linked on the X chromosome. This X had to arrive from his mother. That doesn't guarantee that she's colorblind. She could be a carrier. So you're going to start to recognize things like, I don't know what that second, I'll just put a little dash there because I'm not really sure what she is from this information, but start being able to put together where these chromosomes are coming from. That will make your life easier when we get to the next thing, which is pedigrees or family trees. So if you look at a male, say that this father now has a child. If he has a son, his son will not be necessarily impacted by colorblindness because of the fact that the Y chromosome is going to be passed down. But he will automatically make any female that he has in a daughter has to be passed down to her. So there is kind of this follow through. If a father has some type of dominant sex-linked trait, there are some of them out there that they might mention just to kind of get your understanding. If a male suffers from some type of sex-linked dominant trait, every single daughter that he has is going to be affected as well. Thinking about a father passes an X chromosome down to a daughter and a Y chromosome down to his son. So this is just foreshadowing kind of stuff where you have to start thinking about it. But for a Punnett square that you would have to do, just recognizing that notation is probably perfectly fine. So here's an example. I'll kind of put one that's not a human example that they do. It's a fruit fly example. It says eye color is carried on the X chromosome. So that's their indication you're looking at a sex-linked trait. The allele for red eyes is dominant over recessive white eyes. So this says we have to create a key. So I'll do, if you want to do the key with the X, you're perfectly fine. You can do that. There is a capital and lowercase because there is a dominant scenario. It says cross a homozygous red-eyed female. So that means she, this is the male, they switched it around here. Female would be X big R to a white-eyed male. Males will never have the term heterozygous or homozygous because since they only have one X chromosome, their phenotype reveals what the allele should be. So even though this is a recessive phenotype, they don't have to even do that. Even if it was a red-eyed male, they wouldn't have to say homozygous or heterozygous like they do for her. So this is your setup, and there you go. So this would be a red-eyed female, red-eyed female, red-eyed male, red-eyed male. So you still have to do 2 red eye female, 2 red eye male. All right. If you have any questions, let me know.